Hello, everyone. I'm so excited to get to share my research project with all of you. It's titled The Effect of Indigenous Microbial Inoculants on the Pathogenic Resistance of Ohia lehua to Myrtle Rust. And I know that's a mouthful, so we'll get into what everything means. I was mentored by Dr. Nicole Hinson. I was first introduced to the idea of rapid ohia death by the beautiful ohia lehua trees that lined my backyard in the Big Island as they slowly passed away due to this fungal pathogen. I realized that this fungal pathogen is a deeply detrimental issue to the island of Hawaii, and so I decided I wanted to research more into ways that I could reduce the incidence of this pathogen. So plants indigenous to Hawaii suffer due to fungal pathogen colonization. For example, rapid ohia death, which is caused by Ceratocystis leucoohia and Ceratocystis huliohia. It caused detriment to the native Hawaiian forests as ohia lehua makes up a large percentage of the biomass in native Hawaiian forests and has also been identified on Oahu. So it is a very relevant issue. So what can cause this increased pathogen incidence in native Hawaiian forests? Reduced microbial diversity might be the answer. Reduced microbial diversity is often associated with increased pathogen damage. This can be illustrated by this diagram created by Stroher in a study in 2019, and it found that healthy microbial communities and healthy plants are very diverse and help to reduce pathogen incidence. However, disease communities have much less microbial diversity, and this can contribute to having weaker responses to pathogens. And you can see that the healthy plant has a lot of different microorganisms that are in its environment, whereas the disease community has a lot less diversity. So indigenous microbial communities are inoculated in an environment which needs enrichment. That is the indigenous microorganism methodology. The plant microbiome is made up of so many things, from the air microbiome to the phyllosphere microbiome to the endophytic microbiome. And so I was specifically focusing on the foliar microbiome, which is the microorganisms that live in and on the leaves of an Ohia lehua plant. And so helping to increase the diversity in a microbial community can help the plant have better pathogen tolerance. So it is hypothesized that a microbial inoculant created from wild, healthy Metrosideros polymorpha leaves, or Ohia lehua leaves, contains a host of microorganisms that can help contribute to pathogen resistance. Myrtle rust is the antagonistic pathogen that I use in my research. Although it is not rapid ohia death, I decided to use myrtle rust as it is much more accessible at a high school level research. Rapid ohia death is very locked down due to the virulence of it. So this is another fungal pathogen that is a significant problem for native Hawaiian plant species, particularly Ohia lehua. This image shows the example of effects of myrtle rust on an Ohia lehua tree. It can cause defoliation and eventually death if it is left alone. Previous literature also supports the use of indigenous microorganisms to increase pathogen resistance. Dr. Kamachak, who was a great inspiration for me and helped me perform my experimental design, he created this study where he tested a variety of different treatments on a endangered Hawaiian plant. He used Eugenia koolauensis and determined the effectiveness on its pathogen resistance to Ostroplexinia siti, which is myrtle rust. And he found that the only treatment that statistically significantly decreased Ostroplexinia siti pustule formation was that of a leaf homogenate treatment, which is a source of indigenous microorganisms. Dr. Cameron Egan of University of Hawaii also found that restoration of the microbiome, or specifically the fungal microbiome of the endangered Hawaiian mint, increased its resistance to common powdery mildew. So from previous literature, we can see that there is evidence of this methodology having deep implications on increasing pathogen resistance of specifically native Hawaiian plants. So my research question is, how can an indigenous microbial inoculant aid the native Hawaiian Ohia lehua develop pathogen tolerance to the fungal myrtle rust, or Ostroplexinia siti? Under this umbrella, I had two research goals. First and foremost, I wanted to utilize a controlled experiment to examine the effects of indigenous microorganisms on developing the pathogen resistance of Ohia lehua. My second research goal, after I found out, spoiler alert, that there was a significant difference in the amount of pathogen resistance in indigenous microorganisms treated plants, I wanted to obtain sequencing data to identify a possible mechanism for this pathogen resistance. So under my first research goal, the controlled experiment consisted of collecting healthy wild leaves from the southern mountain range of Oahu and creating a leaf homogenate. So this is where approximately the coordinates of where I collected my healthy wild leaves. And then I took the leaves back to the lab, and instead of surface sanitizing the leaves with ethanol, as previous literature had suggested, I left the outer leaf microbiome intact, as I thought that this could have deep implications on the pathogen resistance of Ohia lehua. So the leaves were homogenized, and this became the treatment or the leaf homogenate and the source of indigenous microbial organisms, whereas half of it was filtered through a 0.1 micron Steravex filter in order to remove as much fungus, bacteria, eukaryotes, etc., to make the control treatment. And this methodology was used to control against any things such as nutritional capabilities instead of, for example, using sterilized water as my control. So that's how I created my treatment and my control groups. So one group of young Ohia lehua seedlings, var glaberrima, which were donated from the Oahu Army Natural Resources Program, was treated with the leaf homogenate or the source of indigenous microorganisms, and the other half were treated with the control or the sterilized leaf homogenate. 72 hours later, the fungal pathogen Osteopuxinia city was introduced to the treatment and control groups at an equal rate. The Osteopuxinia city spores I used were isolated from dried Metrosideros polymorpha leaves that had been infected with this pathogen. And as it was from a dried leaf, we wanted to ensure the virulence of these spores. So we treated them with trypan blue and looked under a microscope. The yellow spores indicate that they are healthy, whereas the blue spores indicate that they are no longer viable. However, over 50% of the spores were viable, so we decided to continue on. Before and after the pathogen was applied to the leaves, randomly selected young Metrosideros polymorpha leaves were imaged, and then ImageJ software was used to analyze the lesions on these leaves. So the pixel input of the leaf itself versus the pixel input of the lesions, the area of the lesions, was put into ImageJ software and was able to get a percentage of the leaf covered by Osteopuxinia city lesions. So this served as my main source of data for the controlled experiment. So if you're thinking about pathogen resistance, if a plant is more pathogen tolerant, it will have less of the leaf covered by pathogens. And so we have percent of leaf covered by lesions, and we can see one group, it has much more of the leaf covered by lesions, and that is of the control. This was the filtered leaf homogenate, which did not have indigenous microorganisms added to the control group, whereas the treatment group has much less disease incidence on each of the leaves. So the mean percentage covered by rust lesions in the treatment group was 12% less than the filtered control, which was shown to be statistically significant using a one-sided t-test, as the p-value was less than 0.05. This shows us that we are aligning with our hypothesis that indigenous microorganisms and increasing the microbial diversity of a plant's environment can indeed help it develop pathogen resistance. So for my second research goal, I wanted to try to dive into the black box a little bit, which is what microbiologists call the plant microbiome. So I decided to use DNA sequencing. DNA from the microbial inoculant was extracted, amplified at the bacterial 16S gene, and sequenced using the min ion. And so one limitation of my research is that I only used a bacterial primer and was only able to amplify bacterial DNA specifically. So I don't have as much information about the fungal or eukaryote DNA that might be in my treatment group, but I decided to dive into what I did have, which was the bacterial DNA. So this is some of the equipment I used. DNA concentration was quantified using a nanodrop. The 16S setting was used on the T100 thermal cycler, and the min ion helped me to read DNA signatures. Through sequencing, Sphingomonas was found to be the most common genus present in the treatment group. This is a pie chart of the percentage of the top 10 genuses identified in the treatment sample, and Sphingomonas is this big blue slice of the pie that we're seeing. So it was a very large amount of the cumulative reads on the treatment sample. So I wanted to look into it more, as it had a much greater percentage of Sphingomonas than did the control when sequenced. Sphingomonas strains have been shown to reduce the effects of phytopathogens. Sphingomonas, in a study by Ina Rebner et al. In 2011, was found to statistically significantly reduce the pathogen incidence of different plants. And they specifically found that Sphingomonas strains isolated from a plant's phylosphere or from a plant's microbiome were the types of strains that helped increase pathogen resistance. However, Sphingomonas strains that were isolated from, for example, the air was not effective in increasing pathogen tolerance in plants. So it's really important, this indigenous microbiome methodology, to use the microbiome that exists in healthy wild plants. In conclusion, the 12% difference in mean lesion coverage between the treatment and the control group, as well as the increased presence of Sphingomonas, supports the hypothesis that indigenous microbial inoculants indeed increase pathogen resistance. Future research is needed, of course, to better understand the many practical applications of indigenous microorganisms. Indigenous microorganisms have been used for so many things, from pathogen tolerance to biocomposting to biofertilizers. So this microbial community is so important in a variety of different aspects. And so diving into that can be very beneficial for a range of different industries, but specifically for restoration of our native Hawaiian forests. Indigenous microorganisms have been used to increase the growth and productivity of native Hawaiian plants, as well as, of course, increase the pathogen tolerance. So finding a way to implement this on a larger scale and in the real world is deeply important to being able to harness the microbial communities that we have on Hawaii. The indigenous microbial inoculant methodology has deep implications in protecting Hawaii's rapidly decreasing biodiversity. Ohia lehua is deeply important to our native Hawaiian forests and is a keystone species. And this methodology, being able to identify that pathogen tolerance is increased by more diverse microbial communities, also lets us know how important protecting these microbial communities are. These microbial communities exist in nature, and it is important that we are preserving our native Hawaiian forests so that we have the richness of these microbiomes to continue using. And it is also a great method to increase the plant health in restorative purposes. So I learned that passion really does lead to motivation. This is some screenshots of what my camera roll looked like throughout the process of being in independent research. So you want to choose something that you are okay with your entire camera roll looking like. That's what I learned. Also, be really confident in your work. You know what you're talking about, and always be willing to learn more and give feedback or take feedback, because people have such wonderful insights and are really wanting to help you and bring your project to the best it can be. I want to thank Dr. Nicole Hinson, who was my mentor. I really could not have done it without you, and I'm so grateful to all the insights you gave me. Dr. Danielle Yogi, your support was really helpful in the logistics of everything, and I am really appreciative of you. Dr. Kamachak gave me wonderful feedback and was also a really great resource, as his paper really inspired me to do this research project. Miss Kobayashi, thank you so much. All of your daily encouragement and support really kept me going throughout it all, and this has been a very informative last 8 months. And Dr. Chan, thank you for being my advisor all the way since the K Fellowship. You really inspired me to love research, and I'm so grateful I have that, and I will be taking that with me as I move forward. And Mr. Tim Chambers of the Oahu Army Natural Resources Program, thank you for your generous donation of the Ohia lehua as well as the myrtle rust sample. And to my friends and family, thank you for being a wonderful support system. Thank you so much. Here are some of my sources. And if you have any questions, please let me know.